So just want to say thank you ever so much for everybody joining us for Invasive Species Week. It's been a good week so far, and lots of interesting presentations. So we're going to be talking about tackling the spread of marine invasive species. We've got, like Sarah said, some great talkers. So Rand is a senior marine ecologist with the APEM Island office in Cork, and Randall spent more than 10 years before joining APEM carrying out marine field surveys around Ireland and other parts of the world. Katie O'Shaughnessy is a senior marine invasive species scientist at APEM, and Katie's got over 10 years experience in surveying for marine invasive species. And then we have Chris, who's a principal marine scientist in APEM's marine laboratory. Chris has over 9 years of working on marine non-native species. Well, 9 years working in APEM, sorry, and 20 years working on invasive species. I lead the invasive species team within APEM. I've worked on invasive species for about 22 years. Prior to working for APEM, I used to provide advice to government. So moving on to the next slide. So just a really quick overview of what marine invasive species are, also commonly referred to as INNS. So typical examples include Crepidula faunocata, the Chinese mitten crab, and sargassum. So invasive species, or non-native species, are species that occur outside of their native range, have been introduced by human activities. And this, the rate of introduction of invasive species has been increasing over the years. So basically, there's been a sudden increase since about the 1700s of the increase in non-native species being spread around and establishing in new environments. Some of these species have environmental, social, and economic impacts, and these are referred to as invasive. So if we move on to the next slide, basically the invasion process can be summarized very simply within the diagram provided. So this is where there's a species starts at a particular point of origin, is transported to a new area, is introduced, becomes established, increases and expands in its distribution, and starts having an impact. And management can be applied at different stages within this process. Now, the Convention for Biological Diversity introduced a hierarchy of management or best practice, and the key part of this, the first part, is prevention of introduction. So this is where an ounce of prevention is worth a pound of cure. So really, kind of, this is where most management should really focus on. Other management practices look at rapid response to new introductions and an eradication of those new introductions, and then containment and long-term control of species that have become established and spread. So if we move on to the next. So just to give you an overview of the topic that we're going to be talking about today. So like I said, it's really focusing on the prevention of marine invasive species being introduced and spread, and how we can tackle that. So we're going to be covering 4 topics. So baseline surveys, looking at things like historical records, rapid surveys, risk assessments, looking at pathways assessments, species assessments and asset assessments, biosecurity planning, and starting to bring this together, the information together, to develop actually a plan on how these measures can be tackled. And then finally, how awareness and citizen science can help to really build and operate in relation to all of this. So basically, baseline surveys are really the key point to start with, and they feed into the other elements. But really, actually, they're all very much interlinked, and basically they all feed into each other, as you'll see as we progress through the talks. So basically, I'm going to hand over to Randall, who's going to be talking about baseline surveys for 1, and then I'll be talking about risk assessments. Katie's going to be talking about biosecurity, and then finally Chris is going to be talking about the awareness side of things. So yeah, handing over to Randall.
Lovely. Thank you very much, Paul, and good morning, everybody. So as Paul said, I'll be opening up and starting this webinar by talking about the importance of collecting baseline survey data in order to inform risk assessments and biosecurity planning. I will be doing this by presenting a study we did for the National Parks and Wildlife Service over here in Ireland in August and September of last year, 2021, as a case study to demonstrate our approach to surveying marinas. And I just want to say at this point, thank you very much to David Lyons and the National Parks and Wildlife Service for working with us, giving us this project to do, and also allowing us to present the project here. So next slide, please, Sarah. So recreational marinas, just as a background, contribute quite a lot to the proliferation of non-native species or invasive alien species, as they're referred to over here in Ireland. Mainly because they act as species reservoirs, facilitating the persistence and spread of species by providing hard substrates in the form of floating pontoons and docks, buoys, other marine infrastructure, and of course, the hulls of boats as well. Invasive species in marinas can be particularly problematic, mainly because they foul hulls of boats and increase drag in the water, so problem for users themselves. They need to be regularly cleaned off pontoons, buoys, and other marina structures, as they can increase the weight of these structures and put pressure on the infrastructure themselves. And importantly, they can colonize resident or visiting boats, which may then leave the marina and carry those invasives with them to their next destination. Next slide, please, Sarah. So invasive fouling species also cause ecological damage by growing over and smothering native species, which is particularly problematic for native filter feeding species such as mussels and sea squirts and anemones, which you will see a lot of when you look at any mussels growing on the bottom of pontoons. Next slide, Sarah, please. So so how do we get an understanding of the presence, abundance, and distribution of these invasive species in marinas? One of the most common methods to achieve this is a rapid assessment survey approach. So this is a method of surveying large areas by employing a semi-quantitative approach where target species are searched for and recorded on an abundance scale during timed searches of specific habitats. So in the context of recreational marinas, it usually requires surveyors to make observations of growth on the sides of floating pontoons. And this method has been used in marinas around the world to record and map invasives and document new arrivals. Next slide there, please, Sarah. So their use around the world is evident by a growing number of publications that are coming out using this method. On top of this, in the last decade or so, independent and private contractors and consultancies are being commissioned to carry out these types of surveys in marinas to inform environmental agencies and organizations. Although the surveys are not currently a statutory requirement in most countries, it is now becoming a common strategy for gathering data to develop management strategies such as biosecurity planning and rapid response plans. Next slide, please. So the overall aim of this particular project for us was to understand the abundance and distribution of non-native species in marinas in Ireland using this RAS service. And the National Parks and Wildlife Service commissioned APEM to conduct these surveys to understand this problem. Next slide. So we conducted rapid assessment surveys on 22 marinas around the coast of Ireland. These varied in size from being only one single or a couple of pontoon sections holding very small numbers of boats up to very, very large marinas. So in the two examples here, you've got a little pontoon in Court McSherry in West Cork. So a very, very small, with some visiting boats and some local boats using it as well, or the large one at the bottom, which has over 500 berths, and it's the largest marina in Ireland in Dunleary. So the time spent searching for species ranged in these from 30 minutes up to 2.5 hours, depending on the size of the marina. Before arriving at each site, we would divide up the marina into sections, so that if we needed to relocate a particular species, we'll know in which section to look. It also helped us in designing the surveys to ensure that we covered a good sample across the whole marina. Next slide, please. Next slide, please, Sarah. Thank you. So during the surveys, we searched for target species, which were provided to us in a list by the National Parks and Wildlife Service. But while we were there, we also recorded any other invasives that we found while searching for the species on the target list. We identified non-natives and estimated abundance using the semi-quantitative Sackler abundance scale, which you can see there. This method was developed to support the observation of communities and species and is widely used in the UK and Europe, especially during rapid assessment surveys. The scale can be easily converted from this descriptive scale, ranging from rare to superabundant, to a numerical scale of 1 to 6. All of the observations for this Sackler scale rapid assessment were made from the surface of the pontoons. As you can see there, Katie lying on a pontoon. Not the most comfortable, but it is very, very effective. Next slide, please. So our observations from the surface of the pontoons were supplemented, mainly because this, for us, this was, we wanted to see if we could add to the surveys themselves in any way, or needed some records. So the observations were supplemented by underwater video and photos. We also opportunistically searched any accessible intertidal structure, such as the marina pilings, particularly if we were in a marina at low tide, it was very handy, as well as any niche habitats known to support fouling species, such as submerged buoys or lines that have been left dangling in the water. As you can see, you can get quite a lot of growth on those. We took several scrape samples at each marina to supplement the field observations also, specifically to ensure we did not overlook any small invasives, such as the polychaetes or juvenile stages of invasive crabs living in the interstitial areas of other organisms. These samples were sent to the Marine Biolabs team for processing. Next slide, please. Overall, in the 22 marinas, we observed 8 species from the target list and found an additional 17 species for a total of 25 invasive species, with Darwin's barnacle, Astrominius modestus, being the most abundant. And as you can see, we were able to gather more data by recording all non-native species observed rather than only those on the list. It was better to record everything we could see. Next slide, please. The largest proportion of the species found were Ascidians, followed by macroalgae and bryozoans. So you see that's the breakdown of all the different species we found, families. And then looking at the geographical distribution and abundance of invasives around Ireland, the greatest number of non-native species recorded at one marina was 15. And the areas supporting the highest non-native species richness being Dublin, which I guess is unsurprising, given that's probably where the most of the traffic also is. Next slide, please. So just to summarize, just that brief introduction to how our methods and how we carried out that. The RAS method is an efficient method for understanding abundance and distribution. We had a total of 25 invasives found in the Irish marinas, and this can be very valuable data to be collected when recording all species rather than just the target list. Next slide, please. So that covers the first step of the 4-step approach that we're going to cover in this whole webinar. If you have any questions, please put them in the question and answer as Sarah has said, and we'll deal with those at the end. But for now, I'm going to hand it off to Dr. Paul Stebbing, Dr. Paul, who will discuss the risk assessment phase.
Brilliant, thank you, Randall. Yeah, I'm going to be talking to a little bit about the risk identification and assessment. So giving a very broad overview of the kind of different types of approaches and the subject as a whole. So if you move on to the next slide. So what is risk? So in simple terms, really risk is the possibility of something bad happening in relation to a particular hazard. So risk can be looked at as a combination of the likelihood and the consequence of actually that happening. So where likelihood is how much chance there is of something bad happening, and consequence is how bad will it be when something goes wrong. So this can be presented as a simple matrix. So on the right hand side, you can see that we have likelihood or probability along the bottom, and then consequence along the left-hand side. So basically, you can color code then by looking at scales within the those two parameters. So basically, you can see on the left-hand bottom side of the diagram where there's low probability and low impact. The person there will most likely be able to jump across the gap, and if they don't, then the impact will be relatively small because they'll fall in water, in comparison to low probability and high impact, where the person there will most likely be able to jump the small gap, but if they do, they'll plummet down onto those nasty spikes. In contrast to high probability, low impact at the bottom right, where the person is unlikely to make that really long jump, but the impact will be low because they'll fall into nice water, in contrast to high probability and high impact, where they're unlikely to make the long leap, but and then they'll plummet to their doomly demise on the spikes. So this can be used to assess and identify events that need managing to reduce risk, either by reducing the likelihood of something happening or the potential consequence of it happening. And this is really an integral part. So risk assessments and looking at risk is an integral part of the process of identifying and prioritizing actions to reduce the introduction and impact of invasive species. So if we move on to talk about how this relates to invasive species in a little more detail. So this is a gross over simplification, but basically you can argue that risk assessments fall into two broad categories. One looking at likelihood and the other one looking at the consequence. Likelihood, for example, would look at the frequency of a pathway, how likely the invasive species are to travel along that particular pathway. So, for example, if you have a shipping going from point A to point B, it would look at actually how many or what propagule pressure of invasive species may be present along that pathway. So that may be dependent on particular characteristics of the particular species being looked at, but also how frequently ships will move. Other types of likelihood assessment may look at operational activities, so for example building of a new port, or particular geographical locations such as marinas, like Randall was talking about. Consequence assessments tend to look at more species related, so what actual impact the species will have. So for example, those that are already present or just arrived, or even horizon scanning. Like I said, this is a gross over simplification, and that most assessments tend to be a combination of, or at least contain elements of both. An example of this is with the GB non-native species secretariat's risk analysis process that incorporates several stages of risk assessment. So this is the diagram on the right-hand side. So they incorporate horizon scanning as well as risk assessment for species, which encompasses looking at the likelihood of entry, establishment, spread, as well as impact, although it's specifically looking at species. So now I'm going to move on to provide some examples of different types of risk assessments. So this one, looking at pathway risk assessments. So pathway risk assessments tend to cover a single specific pathway. So so the approach tends to be specific to that actual pathway, and really take a deep dive into assessing actually the elements that would affect the movement of invasive species along that particular pathway. I was going to use ballast water as an example. Ballast water is a known and highly and recognised means of transporting or movement of invasive species in the marine environment. So just as a recap, ballast water is where water is taken up onto a vessel when it's unloading cargo, and then that ship will move to a different location, and the water will be then discharged, and with the uptake and discharge, the potential movement of non-native species. Ballast water is an essential part of ship operation to maintain the ship being upright, basically, as well as momentum. So really, there's two different types of assessment that are used in relation to looking at ballast water. So there's a species-specific risk assessment, whereby the risk of transfer of a particular species is looked at, and this is looked in relation to, for example, the physiological environmental limits to survive the pathway and introduction. And this is a very effective way of looking at a small number of species, but it requires an awful lot of data and information actually on those particular species. In contrast, there's the environmental similarity risk assessment process, which compares the physical condition at either end of the pathway, and how the dissimilar environment basically can present a poor likelihood of survival, while very similar environments can present a high risk of survival. One of the issues with this particular process is that it can actually miss species that are highly adaptive to environmental conditions. So it doesn't present a necessarily as a robust process, but it's a lot easier to get the actual information available to be actually do the assessment with. So moving on to looking at assets and operations risk assessments. So this is a type of risk assessment that we apply a lot within APEM. So this looks at assets which tend to be a specific location. So for example, a marina, or looking at operations that tend to focus on a specific event. So for example, a construction of a port or a particular event, sporting event. So basically, this looks at a combination of qualitative and quantitative information, or a particular approach that can be taken in relation to these assessments. So the approach that we tend to use is looking at all relevant pathways identified within the actual area or in relation to the operation. So, for example, within marinas, that could be large yachts and going down to the smaller vessels, as well as actually particular features within the marina as well. So we look at the frequency of pathways, for example, how often the yachts entering the marina will come in and out, and where they actually go to as well, and basically score these, provide a point score. We also look at the invasive species at a particular site, so using methods that Randall described, and provide a score in relation to those particular species. We'll also look at horizon species, which I'll talk about in a little bit, and look at actually how so horizon species in relation to those that are in near vicinity, or those within the actual the country, and or those actually horizon to the country itself. So through that, we can actually provide an overall score in relation to the potential risk an asset and operation may present. This is a highly flexible approach, and it can determine the risk of introduction and spread out of a particular asset or in relation to a particular operation. And this can also, because it's comparative and very repeatable, it can be used to determine hotspots if applied to multiple sites. So I mentioned horizon scanning. So just moving on to that. So horizon scanning is looking at the consequence of something coming along. So it's looking at, in this case, looking at what's next, what species is likely to arrive next within the UK. Or the country being studied. So in the UK, the GB non-native species secretariat run a process for horizon scanning. I think it tends to be approximately every 5 years or so. And this uses a consensus method using technical experts. So you have large groups of experts who will basically provide an initial ranking score for species that have been identified as potentially arriving within the UK in the near future. So these scores look at arrival, establishment, the magnitude of potential negative impacts on biodiversity, ecosystems, human health, or the economy, and then these are scored from high to low, and then provide a cumulative score is provided for each species, and then basically through a discussion and review, then a common consensus on the scores are provided. So the last event horizon scanning process within the UK was in 2019. Reviewed about 250 odd species, and then through this process we came to a final conclusion of about 100 species, and then there's a top 30 that was published on the non-native species secretariat website. So Helen Roy from CEH manages this whole process for the non-native species secretariat. So you can see on the right-hand side the marine species that are suspected to arrive in the UK in the near future, taken from the top 30 on the GB non-native species secretariat website. So moving on to the next slide, and talking a little bit more about species risk assessment. So this is a key tool used in relation to actually trying to identify what risk a particular species presents. The difference with this and horizon scanning is that it's normally applied to species that are already present or that have been that are newly arrived within an area. So the GB non-native species secretariat again uses this on a regular basis, and basically they have several different types, but the rapid assessment that they use looks at entry, establishment, spread, impact, and climate change. So you can see on the left-hand side an example of how entry is assessed. And so basically there's a scale of response of how likely the reviewer would look at entry and how confident they are in relation to it. There are other species-based risk assessment tools available. So, for example, ASISC, which was developed by CFAS and maintained by CFAS still. So ASISC comprises a list of 49 questions on the species' biogeographical, historical traits and its biological and ecological interactions. It also has questions on socioeconomic impact and ecosystem services, as well as a section for climate change. And so it provides a quantitative scoring mechanism in contrast to what is more a qualitative scoring mechanism presented by the non-native species secretariat. So moving on, I'm going to pass over to Katie to talk a little bit more about biosecurity planning. So thank you, Katie.
Thanks, Paul. Hi, everyone. I'm Katie O'Shaughnessy, and I'm part of APEM's invasive and non-native species team, and I'll just be talking for the next few minutes about biosecurity planning. Next slide, please. Okay, so at the expense of perhaps showing this diagram too many times, I just wanted to point out where we suggest biosecurity planning sits in the overall invasive species management scheme. So you'll see it's kind of the glue connecting all phases, as shown by the arrows here, as there is often overlap or interactions to some extent with all other parts of invasive species management. So for example, baseline survey data do feed into biosecurity planning, but biosecurity measures put forth within a plan should actually include the implementation of baseline surveys if they haven't been done yet, as well as continued monitoring. So in my humble opinion, and not just because I'm presenting on it, I do think biosecurity planning could really be considered the cornerstone of many invasive species management schemes. Next slide, please. Okay, so as many of you probably know, Ben Franklin was famously quoted saying, "An ounce of prevention is worth a pound of cure." And seeing as though I am American and I understand the imperial measuring system, I can translate this for you in the context of marine invasive species. So as you can probably tell, this means that once an INNS is established in a new area, they are nearly impossible to eradicate or control. So preventing the introduction is recognized as the most effective and least environmentally damaging intervention. Therefore, biosecurity plans identify realistic, pragmatic, and cost-effective procedures that reduce the risk of species introduction and establishment. And these need to be highly specific to the characteristics at the site, but there are general measures that will likely apply across all sites. Next slide. So typically, at a minimum, these plans include an introduction setting out purposeful aims and objectives, which should include scope such as where and to whom the plan applies, a section identifying the risks that the biosecurity plan will cover, for example, either species, pathways, or both, which was just discussed by Paul. Then the measures by which the risks will be addressed, which are usually in the form of general or specific actions to be taken, steps for how the plan will be implemented, including responsible parties, and then, of course, a review process identifying who the plan will be reviewed by and how frequently. Next slide, please. Thank you. So generally for marine biosecurity planning, the following actions can potentially be put forth as measures by which the risks will be addressed, but these are simply examples and by no means an exhaustive list. So first of all, recommendations might include appointing a general biosecurity manager who covers the entire geographic area of the plan. So this person should be responsible for, say, organizing workshops, trainings, holding regular meetings with stakeholders, as well as orchestrating all general biosecurity matters for the entire area. Next, perhaps appoint, thanks. Appointed biosecurity champions specific to smaller sites within the entire plan area. So this person should be responsible for any invasive species and biosecurity related issues and activities at a specific site, such as a marina. So by nature, there may be several champions within one area covered by a biosecurity plan. For many areas, it may be recommended to install washdown facilities to facilitate removal of biofouling invasive species from boat hulls and equipment if none are already available. And then for other areas, perhaps modification to existing haul out and washdown facilities might be needed to ensure the ability to catch wastewater so it does not wash back into the sea. It may also be recommended to stay up to date with national horizon scanning reports put out by the GB Non-Native Species Secretariat. Next slide, please. So also, additional and/or heightened biosecurity measures during events should be implemented, as these pose a risk to invasive species introduction and spread via increased number of people and equipment on site, many of which may actually come from distant regions, bringing in non-native species. It may be necessary to develop standard procedures for monitoring high risk and horizon species. Again, an action that may come from pathway and species risk assessments as covered by Paul earlier. To ensure INNS are correctly monitored both now and into the future, biosecurity plans should recommend species ID and monitoring training for local staff and volunteers. And then finally, the backbone of every good biosecurity plan should include the implementation of biosecurity and invasive species awareness campaigns, such as the Check, Clean, Dry. Next slide, please. As many of you are probably aware, these biosecurity campaigns are aimed at the public and are now becoming more common around the world, as demonstrated here with the various signage from the US, the UK, New Zealand, and there's one there with Canada. These campaigns, such as UK's Check Clean Dry or North America's Clean Drain Dry, aim to educate regular water users about the way in which aquatic plants and animals can unintentionally be transported by equipment such as boats, kayaks, fishing gear. And they provide information about how invasives threaten local ecosystems and economies. So really putting it into context. Next slide, please. However, despite even the most robust biosecurity measures being in place, we all know that species may still arrive to an area. So it is essential to have some sort of early detection and rapid response plan in place. So early detection and rapid response, or EDRR, is defined as a coordinated set of actions to find and eradicate potential invasive species in a specific location before they spread and cause harm. So early detection involves identifying pathways of potential introduction and tailoring monitoring schemes to focus on hotspots of introduction or movement to increase chances of finding new arrivals early on before they become established. Now, rapid response plans are put in place to take urgent action to eradicate, control, contain, or manage any new incursions of an invasive species. And I think this diagram from the U.S. National Parks and Wildlife Service demonstrates the importance of EDR, nicely showing the priority of actions for invasive species management, with prevention requiring the least amount of time and cost, and then on the other end is long-term management as the most costly and time-consuming. Next slide, please. In order to prepare for rapid response, a rapid response plan is required. So this plan is a document that presents a framework for managing new incursions by providing clear, proactive, coordinated actions to undertake. Many biosecurity plans do have a rapid response plan embedded within it, as rapid response is often seen as a part of good biosecurity planning. So these plans should have in place details of the stages of response with associated communication and responsible parties so that actions to eradicate, contain, or manage an incursion are dealt with swiftly, as you can see here in this diagram. So communication involves knowing when to alert national and/or local authorities, as well as internal comms to alert and advise members of staff, as well as external comms to alert stakeholders and the public. Now, responsible parties need to be prearranged and could include an organizing response group, as well as an operations group, which would implement actions on the ground. Now, these plans obviously need to be bespoke to the site, its pathways, and the owner of the plan, but can follow a general structure like the one you see here. Next slide, please. So overall, biosecurity planning requires an understanding of pathways, current species present in the geographic area, horizon species, and any proven treatment and control measures that might be available, although we all do know that this is quite difficult in the marine environment. These plans should also include early detection considerations and rapid response. And I think we should all consider linking up work being done on a national level to ensure we capture what works, what doesn't, perhaps realize any knowledge or geographical gaps, and also essentially so we don't have to reinvent the wheel every time someone wants to develop one of these plans. I think perhaps most importantly, these plans really won't realize success until there is collaboration among multiple parties. So involving invasive species biologists, ecologists, innovators, perhaps shipping professionals, watercraft clubs, local and regional authorities. These are all examples of parties that might need to be considered when developing one of these plans. And this is because many of these parties, especially the locally based ones, will be essential in helping gain stakeholder buy-in and public uptake of the plan. Next slide, please. So with that being said, there have been several biosecurity plans published and publicly available in the UK, and dozens more across the globe, which are specific to certain geographic areas, habitats, species, and/or pathways of introduction. And APEM has actually developed several biosecurity plans over the last few years, covering biosecurity for construction and aquaculture operations, ports and marinas, and conservation areas such as SACs. With that, I will conclude and say thank you for listening, and I will now pass the torch to Dr. Chris Ashleby, who will talk about invasive species training and awareness. Thank you.
Yeah, thank you, Katie. As Paul mentioned, and I'm Chris Ashby, work in APEM's Marine Lab. I'm just going to cover this section on about raising awareness and training. So, I mean, as we all know, monitoring for non-native species is like an ongoing process, and as Katie and Paul previously mentioned, all the stages are interlinked, and none of the previous sections that we've talked about are considered an endpoint in themselves. So once you've conducted your baseline surveys and done your risk assessments and developed your biosecurity plans, you're moving into a phase then of continual observation and monitoring. And the purpose of this phase is really to detect changes in either species composition or distribution of the species known to occur, and to try and determine if there's any opportunities to prevent any further introductions. So to do this effectively, really needs to be a degree of education and training to stakeholders and users about the outcomes of those previous phases. So the outcomes of the baseline surveys and the risk assessments and the biosecurity plans, and how you disseminate the information, try to engage people in that process will depend on situation. It'll depend on the skill level of the people you're trying to talk to. The methods that you want to use they also sort of provide the opportunity at this stage to incorporate citizen science, or perhaps tap into other schemes such as shore search surveys that are ongoing by local wildlife groups, for example. And by using those avenues, it can really sort of help increase spatial and temporal coverage of the surveys, as well as potentially being a cost-effective monitoring method. So firstly, as we have here, biosecurity awareness and training. Firstly, if we've done biosecurity plans or conducted baseline surveys and have reports that we can share, sharing those and making sure that relevant stakeholders are receiving those. Make sure that people are aware of the outcomes and your recommendations. Then we've got signage, obviously, as Katie's also mentioned. That putting signage in a marina, for example, about biosecurity measures can really help increase the buy-in with conducting the biosecurity measures. Although the sort of check, clean, dry motto we've all become accustomed to, it's possible that specific risks might require bespoke biosecurity measures. So it might be necessary to train people in non-standard methods. For example, you might require people to soak equipment in fresh water or in disinfectant baths. So you really need to ensure it's being done correctly, and that people also, any biosecurity training must include highlighting the potential consequences of not following the methods. The what if scenario. For example, does the species have the potential to spread associated diseases? What are the potential ecological or economical consequences of allowing the spread of the species? It's sad to say, but people will be more likely to buy into biosecurity if you highlight the economic grounds, because it's something that directly affects them. So the next slide, please, Sarah. So for the monitoring itself, it's likely you're going to require some degree of survey effort, whilst incorporating experts such as APEM. It's we're probably most qualified to do the work, but having us at every location isn't practical. So this is where the training of citizen scientists could come in. Really, what we're trying to do here is encourage vigilance for newly arriving species, monitor the spread of existing species. So by using properly trained citizen scientists, and possibly the people who are already working and using the areas, you can expand the geographic range and temporal extent of the monitoring in quite a cost-effective manner. So it might be that we you could run a workshop, for example, training on survey methods that don't require any specialist equipment, such as the rapid assessment surveys that Randall discussed, or the abundance, distribution and range technique. You could also provide training for people to take scrape samples, for example. Scrape samples of fouling communities can provide lots of records of non-native species, and they can be relatively easily incorporated into maintenance or inspection schedules for offshore infrastructure or marker buoys or the dry docking of vessels, for example. The samples are going to be sent to the lab for identification, but as I said, it does save the cost of having a dedicated sample collection team on site, and it can be done with relatively little training, and in a very quick and efficient manner. And finally, I mean, you could, for example, to train people to deploy and retrieve sediment panels that can likewise be sent to the lab for analysis after a period of colonization. So we'd like to move on, Sarah, please. So identification training, that could involve either in situ, so in the field identification, or in the lab. It would be relatively easy to train sort of non-specialists to recognize certain species, you know, for example, wakame, Undaria pinnatifida, wireweed, the Sargassum muticum. They're all quite easily recognized, and having citizen scientists looking out for these larger, more recognizable species can help provide an early warning of their occurrence in an area. But it's also worth noting that many non-native species are the smaller invertebrates, which do require more specialist identification skills and a good knowledge of the similar native species. For example, often when you find a non-native species, the first step in discovering it is looking at it in the context of the native ones and thinking, that doesn't look quite right. It doesn't quite fit any of the descriptions of the native species. So what could it be? But even then, there's varying degrees of skill levels amongst different labs in recognizing non-native species. As part of APEM's remit in running the NMBAQC scheme, we have conducted a couple of ring test exercises that are focused on non-native species, and the results of these show widely varying degrees of how well different labs recognize non-natives, and especially for those which are less distinctive or horizon species, so not yet found in the UK. But similarly, as part of the scheme, we run workshops already for beginner, intermediate and expert level analysts, and we could conceivably run a workshop focused on the identification of these smaller non-native species to help raise the awareness and recognition amongst other scientists, and hopefully people will be recognizing those in their routine samples, not just in the samples that have been specifically collected as part of non-native species monitoring programs. Just move on, Sarah, please. And then it might also be a case that the training isn't physical training, but it could take the form of bespoke training identification materials. There is already a really good photographic guide on some of the larger species of concern in UK waters produced by the MBA in conjunction with Natural England and Bromley Trust. And you could conceivably make a more targeted guide for specific areas of regions of the UK based on the outcomes of our baseline surveys and the risk assessments. It can cover then the species that occur in the area from those baseline surveys, but it's essential it also incorporates horizon species that are not present in the area of interest, because those are the ones that we want early warning of if they occur. Species fact sheets can be a useful tool for raising awareness of specific species of concern. If any of you saw the presentation yesterday by Hannah Tidbury of CFAS, she presented a good example there of a species fact sheet for American lobsters. Similar fact sheets are available for other species, or could be produced for those that they don't already exist for. And finally, we can produce training materials for different survey methods. So they could be disseminated, for example, to marina operators to allow people who in the marina to conduct rapid assessment surveys, for example. I mean, overall, really, the key to raising awareness and planning effective training is to adopt a flexible approach. It needs to address the key pathways identified by the pathway risk assessments, and it needs to be tailored to the skill levels and interest of the attendees. So I think that concludes the webinar. I'd like to thank you all for your time listening, and I hope you've all found some the information useful, and any questions.
Brilliant, thanks Chris. We do have a couple of questions in already. So the first one, anonymous, is there any research into how we can reduce ballast water spread of INNS? Potential management strategies to reduce the chance of spread from ballast water. Who wants to take that one?
I'm happy to pick that one up, Sarah, if you want.
Thanks, Paul.
Okay, so there's actually a ballast water convention that was introduced by the International Maritime Organization. So the ballast water, and sediment management. So it actually came into force on the 8th of September 2017. And basically, the thing is that this presents standards that vessels are required to adhere to reduce the risk of the transfer of invasive species as well as pathogens with ballast water and sediment. So ships are required to have a ballast water management plan. There's also a ballast water record book and a certification to show that they've meet certain standards. Now there's two different standards that basically are applied. There's one where basically the vessel has to exchange ballast water in more than 200 nautical miles away from land in at least 200 meters depth of water. And there's another standard where basically they have to have a treatment system on the ship, which will treat the ballast water to the point where basically the water presents no risk. The organisms are basically either dead or will not reproduce as a result of that. So yeah, there is a lot of work going on in relation to the implementation of that that particular convention and making sure that vessels are adhering to it around the globe. So I hope that answers that question.
Terrific, thanks Paul. There's another question from Andrew Want that came in chat. I noticed the image of settlement panels. Is there a standardized model that is recommended?
Yeah, I can answer that. There is, and the model that we demonstrated there actually is the standardized model. I forget the name of the paper, but I can dig that out and let you know the answer to where it's described. But essentially it's the dimensions that are the critical thing, because then it can give you the abundance per meter squared quite easily, and it means that you're sort of standardizing the sampling area each time. The advantage of the one that was demonstrated as well is you might have noticed it had sort of a triangular bit on the bottom. That's partly because of stability within the water column, but it's also partly because different organisms prefer different habitats. So some of them will colonize the inside of that triangular bit because that's more shaded, and others will colonize the sides where they're more in the sunlight, for example. So yeah, there is a standardized model.
Terrific, thanks Chris. Next question is from, apologies for the announcement of your name here, but I think it's Julio. What is the potential for integrating genomic approaches in the detection of INNS and their invasion pathways? Any takers from our team?
I can pick that up if you want.
Thank you, Paul.
Yeah, I think looking at molecular detection of species is kind of increasingly becoming more common in relation to surveys and pathways. So for example, the use of environmental DNA or eDNA. So that's the detection of basically free bits of DNA actually floating around in the environment. The only problem in relation to the use of eDNA, well, it's very technical and quite difficult to achieve in relation to some species, especially with varying levels of the DNA being present in the water, but also it's georeferencing. So actually being able to say that this species is definitely here, especially when you're looking at large tidal ranges, and also the things that DNA can be moved around through other means, or because you're looking at the presence of a particular species and you find the DNA, it doesn't necessarily mean that the organism's there as well. So yeah, I think that it's one of those things that there needs to be quite a lot of interpretation of using molecular genomic approaches, detection of INNS really. But yeah, I think it's one of those things that it's definitely a developing field, and we provide quite a lot of advice on the interpretation of eDNA results.
Thank you, Paul. Dawn's put a question in the chat. Dawn Diamond, are there any barcoding studies being carried out alongside this work?
I believe that there are various barcoding projects being undertaken. It's not something that we are directly involved in. We have supplied samples for some, I believe. I don't know if Chris has any more knowledge in relation to barcoding work on this.
Yeah, I was going to say, I mean, actually, there is some work that's done. There's very few invasive species that are detected solely through genetic work and barcoding. Part of the reason for that is that potentially any species from a suitable environment could be introduced to the UK, and there's so few species in reality that have been barcoded in the world. So it's not like you could find a species, barcode it and get the answer of what it is. But there is also things like recently the carpet sea squirt Didemnum vexillum, which I think probably most people are familiar with now. They did some barcoding work on that and found out there's actually two species that have been introduced. So there's Didemnum vexillum and Didemnum pseudovexillum. And really those do differ morphologically, but they're quite difficult. So I think if you wanted to be 100% certain, you probably would follow a barcoding approach at that point to try and determine which of the two species was present in your area. Okay.
Thank you, Chris. And the last question, but come on people, there's still time if anyone else has got that burning question. How much impact has water temperature, i.e. UK range, been assessed as having on the successful establishment and subsequent invasiveness of an INNS? Does the potential increase in surface water temperatures being seen then elevate the risk of spread of specific species? Oh, crikey, that was a tongue twister there at the end. Who wants to take that one, guys?
I think, I mean, the answer of whether it increases the potential risk, obviously it could increase the risk of a species from slightly warmer waters being introduced. I mean, generally, successful introductions to areas, they come from a similar latitude of the world. So a lot of ours in Britain come from Japan, northern China, or the sort of similar latitudes of the United States, for example. So by the water temperature increasing, then you might start getting things from other parts of the world where it is slightly warmer. However, you would have less risk of than the ones that can only tolerate the cooler waters. And you do find this even within the UK. There are species that have been introduced to the UK and can survive in the south of the UK, but it's too cool for them currently further north. So as sea temperature rises occur, then potentially they will expand their range further north within the UK. So yeah, I mean, I think it's one of the sort of parts of a horizon scanning process really is to look at the physiological tolerances of a specific species and could it survive if it got here. There was the recent occurrence of a lionfish off the south coast, which at the moment I'm not sure it's been 100% verified that it was that it was living there properly, or whether it had been sort of released and caught almost immediately before it had a chance to die. But that species, for example, at the moment, the water temperatures in the UK wouldn't be suitable for lionfish to survive and breed. Potentially in the future, that could change.
Terrific, thanks, Chris. Another question in from Jack Sigmund. Marinas were used in your examples today. Are there INNS that we should be aware of in the open ocean or further from the coast in the UK? And how would you go about surveying, monitoring for these species?
So out in the open ocean, there's a lot of talk of sort of infrastructure such as wind turbines and oil rigs providing what they call stepping stones for species. So a species introduced in coastal waters might be able to, if the larval stage can reach a piece of offshore infrastructure and grow on there, then the next larval stage can move on to the next turbine or oil rig. And so there is evidence to show that the species do cross the open ocean. However, if you were talking in an unimpacted environment, finding non-native species far offshore is not that common. There are exceptions. I mean, things like the American lobster, for example, that can, because it's quite mobile, it will occur where the lobsters would occur. I think marinas have been used quite a lot in the examples today. That's partly because they're an easily accessible place to survey, but they are also, as Randall said at the beginning, they are a hotspot for non-natives. Pleasure boats actually, the hull fouling of pleasure boats is a vector that has led to the spread of quite a few species around. And also marinas tend to be near ports, and as Paul mentioned, ballast water is another large vector. Tend to get less hull fouling on commercial vessels, obviously, because every bit of hull fouling causes drag, and that increased costs for operators, so they're quite good at keeping their hulls quite clean. But they do have other things in ports as well, like sea chests of vessels, for example, can provide crevices where things can hide.
Great, thank you, Chris. Chris, I don't know if you've seen in the chat regarding the last question that we'd had about the impact of the water temperature. The attendee said the DVEX in Kent, i.e. warmer, appears to be more invasive than further north.
Yes, so that could be an example where the water temperature is limiting the invasiveness of the species in the north of its range currently. But if water temperatures increase, then it could cause more of a problem further north.
Terrific. Well, unless there's any final questions from anybody, I'm gonna leave that as an open pause just to see if anything appears. It doesn't look like it. So thank you everyone for attending our INNS Awareness Week webinar on marine species today. I hope you found it informative and useful. As mentioned at the beginning, we'll be sending around a link to the recording later next week, and also details of our next webinar, which I can tell you the title, but I can't tell you the date, I'm afraid. We're still trying to confirm it. But she says, with a drum roll, it's called Water, Soil and Poo. What can eDNA tell you. I hope that as many of you as possible will be joining us for that one. And thanks again to our very knowledgeable and talented speakers today, and have a great rest of your Wednesday, everyone. Bye.